All right, we are starting Honors Research Night on Wednesday, October 24th, 2022, with Jess Kramer as our first presenter. Go ahead, Jess.
I'm Jess, and I represent on the hydrocarbon receptor effect on maturation. How many science people are here? Like bio, awesome. So we're going to do a quick overview of immunology. Your immune system exists to keep you alive. It's going to fight bacteria, viruses, parasites, all sorts of pathogens from trying to kill you, which is nice of it. It also prevents your body from attacking itself, and it helps to preserve the good gut bacteria that you have in your GI tract. I mentioned if you've heard about the microbiome, and there's all sorts of prebiotics, probiotics. Those are actually really important to need all of those bacteria in there. And the aryl hydrocarbon receptor, the AHR, does play a part in that. This is also the macrophage phagocytosis bacteria. We're going to talk a lot about antibodies today. Antibodies are just protein that your body needs to either label the things that it's trying to eat, or it neutralizes them completely. Because if you have like a knife but you put a sheath on it, you can't really stab anything that well. Same kind of concept with the antibody. Or there's a lovely little label on it, so macrophage come along and eat it. So what is an aryl hydrocarbon receptor and why do you even care? It is a receptor on cells and many places in the body, but we're going to focus specifically on gut. It helps to protect the good microbes by limiting the amount of antibodies produced. It's a transcription factor, so that means it's going to bind onto DNA and produce effector proteins to do its job in protecting yourself, long story short. So at the top, kind of where all of those loops of yellow and orange are, that's where ligands going to bind. And then the bottom, you see it kind of looks like a double helix, kind of like DNA does. That's where it's going to bind to the DNA and eventually start transcribing proteins. What we did in our lab, partially what we did, partially what Dr. Dupre has done with other students in the past, is use TCPD, which is 2,3,7,8 tetrachlorodibenzoldioxin, as an AHR agonist, which is a TCPD. I was trying to say that a lot of times. As an agonist. So it is going to like ramp up the AHR activity. What these graphs are showing you is the more TCPD that we treated mice with, the less antibodies that were produced. And there is some recovery at the end of the experiment where even though the AHR is still activated, the amount of antibodies just about levels out. There's also a difference in response for lots of different infections between your blood and your gut bacteria. So we also compared the difference between those. So what we did is partially a proof of concept of experiment. So it's the same concept with vaccines, same concept of getting a cold. The more times you're infected with something, the better immune response you're going to have, right? Same thing with COVID vaccines. That's why you get a booster, more exposure, better memory, better affinity that your antibodies are going to have for the antigen. Affinity just refers to how strong that connection is between the antibody and the antigen. If you're trying to neutralize something, if you're trying to label it to be eaten, you want it to have a pretty strong like hold on there. You don't want them popping off all the time. So ideally, you have a higher affinity. What we predicted is because the AHR has an effect of decreasing the total amount of antibodies in an immune response, we also think that it's going to decrease the amount of affinity that this are going to have as well, and that's what we are testing. To quantify affinity, we're using this thing called an ELISA, an enzyme-linked immunosorbent assay. What you need to know about it, there's a lot of steps that happen, and eventually it produces color change in accordance to like what the concentration is. So you can see on the 96-well plate, there's a really big concentration at the top. There's going to be a lot of color change at the bottom. There's not a huge concentration of antibodies. There's not a huge color change. So when you make a graph that looks like this, you can get lots of information about the amount of antibody in there and the affinity of that antibody. So we also, so group one, if you only have one exposure to an antigen, you're not going to have a ton of antigen-specific antibody for it. If you're exposed to it 4 times, like group 4 on the right, you're going to have a pretty big response. So because of the ELISA, there's going to be a color change when there's a big concentration. There's a big concentration of antibodies if you've had 4 immunizations or inoculations. So it'll be a big color change. We also used a pleiotropic salt called guanidinium hydrochloride that disrupts non-covalent bonds. So if anybody remembers from basic chemistry, covalent bonds are the really strong ones where they share the electrons, right? Hydrogen and ionic bonding are not quite as strong. They're a little bit more easily breakable. And that's where a lot of the affinity comes from. So if your group 4 is a little bit easier to see, like the 2 yellow lines, the top one is a sample from an animal that was not treated with the guanidinium hydrochloride when we were doing the assay, and the lower yellow line with the gray dots was treated with the salt. So we would expect to see a lower optical density of that line that was treated with the salt because it's disrupting the bonds. It's going to cause the antigen to fall off of the antibody. That's exactly what we're expecting to see. This is just a different interpretation of that. There's lots of different statistics you can do on this, but in cleaner form, for 1, 2, 3, or 4 weeks, the top black line is the optical density, or optical density, the area under the curve for the optical density versus the x-axis. Obviously, that's higher than the salt treated one, and we're also measuring like the percent difference. So the percent difference between the salt and unsalt treated lines kind of gives us insight into how many bonds are being disrupted, what kind of bonds they are, lots of molecular geometry for like angles and what things are meeting in the immune response. So we need to do a lot of ELISAs for this. We have to do each sample in triplicate and then do that each twice. And we've got about 200 of those. So we are mid ELISA storm at the moment. This is our preliminary data from our proof of concept experiment. We only had 14 animals for that. The past experiment that was done, there were about like 60. So we don't have any conclusions from the TCDD yet, but that is what we're going to be working on for the next month or so. Thank you.
All right, questions thrown out to all of you.
Yeah, you said you use animals. What animals we use mice. Oh, cool. Yeah. I don't know if any of you are in Ross Hall ever, but there's like the animal research lab in the basement down there. Yeah. Yeah, so there's different isotypes of antibody. They come in different flavors, from a lot of bio professors like to say. Long story short, we focused on IgA, immunoglobulin A. It's the most highly produced antibody, and it's also the most secreted. So your body makes a ton of them, literally just to put into your intestines and move them out. So it protects stuff in your gut. It's a lot easier to measure than other things that are not produced like quite as quickly. Like we could do other isotypes that just wouldn't give us like as strong results.
Are you hoping that this research will accomplish?
Yeah, so gut bacteria has huge implications. It's like kind of an up and coming thing in immunology. It has potential like treatment implications for ulcerative colitis or IBS. If we could like help protect that good gut bacteria by messing with these mechanisms, that could be a potential like drug possibility for those diseases.
Yeah. What's your timeline then for kind of finishing up the research that you're working on?
Yeah, so we're actually applying to NCUR and the Fall Symposium. We're also going to Immunology 2020 in May, and Phi Beta is a biological honor society. They have a conference in April. So we would like to have all of this finished by February. So we're already applying to those things, but to have it all done and solidified by the time we hit kind of conference season. So
All right. And are you graduating in the spring?
graduating.
Oh, okay. All right, well, thank you so much. If there are there any other questions? Okay, thank you so much.
So next is Angie.
Let me move this just a little bit closer. This all. I put it.
Oh, that is much closer. Hello. Oh, great. I get to stare at my face this entire time.
And then that little clicker there should.
Mhm. Oh, that's so fancy. Okay, cool.
All right, so next up is Angie Kaufman. And go ahead and introduce yourself and your topic.
Hi, I'm Angie. I'm gonna make you guys switch gears in your brain a little bit. I am an English major, so I work better with words than with cells and biology and things like that. So on that note, my thesis is Language Dissected, an exploration on the understanding of language and its application to Tolkien's Elvish. What does that mean? So my thesis is on language and why it works and how it works. Because we use language every day. I'm using it right now, and we use it in basically everything we do. But I don't know if any of you have ever wondered this. I have. Why did we do that? Why does it work that way? So we take language for granted. We use it all the time, as I just said, and we don't think too much about it. But there's so many different languages and so many different grammar systems that go with different languages, but they all work. So why? Why do some people function better with German? Why do some people function better with English? Like, why is that a thing? That was something I've been asking myself for most of my life. I always thought I was the weird kid because I was like, why are we calling that a fire truck? Why? Like, what made people think fire truck or fire? Why those words? Why that sound? I thought I was crazy, but here we are doing a thesis on it, so it's okay. I mean, I just like, why do we call things the way that we do? So I'm going to dig deeper into that. And if we understand how language works and why it does, we can communicate more effectively on our day-to-day basis with each other, and also as a global community, whether that's from people in Germany, people in China, people here. If we understand how language works as a whole, we can understand each other in different countries better, and it will get along better, which is always the goal. So there's a big conversation to be had about language. Linguistics is one specific field of language study. It's basically an understanding of how language works and diving into the details of that. And then by understanding how language works and how we use that today, the field of linguistics itself and communication outside of linguistics, so just like how we speak and how we do other things like marketing even could be effective in that. We can advance all of that in ways that will help everyone. And then specifically Tolkien scholarship, because I'm a nerd, it's also a field that is actually steadily growing. It is an actual thing. People do just study Tolkien for a living. Don't ask me how well that living works for them, but it is a thing. And it's a pretty modern one, so that one is new and steadily growing, as I said. So I'll be contributing to that, even in the smallest of ways. So before I go into all the cool things I've learned about my thesis, there's some vocab words you need to know, otherwise you'll be very lost. Morphology is the study of the form of things. So generally how things are made up and how they work. A morpheme is a unit of language that cannot be further divided, so like our suffixes, like ing and ed, those things you can't take apart without destroying their meaning. Those are very important parts of language. And then a phoneme is the smallest unit of sound. So even smaller than the ed or the ing, it is the specific sounds that make up words. For example, like C and P are some, and then there's others like TH and PH that are also phonemes, because you can't take those apart without losing their sound. And switching gears into Elvish, as of 2008, 25,000 Elvish words have been published. I remind you that Elvish is an invented language. It is not something that people speak regularly in a country. It is an invented language made up fully for a book, but there are 25,000 words that have been published in this language. Let that sink in for a second. That's crazy. That's a lot. I don't think there's any other or very few invented languages that have that many words attributed to them. And it's mostly because in Elvish, you have all these systems. You have a grammar system, you have dictionaries that are actually published and purchasable on Amazon. They're a thing, they're like real. And there's websites, there are linguistic forums for Elvish. And that doesn't exist for most other languages, which is another contributing factor to why I chose Elvish as the language I'm comparing it to. Because Elvish is so developed and so modern and vibrant even, that I wanted to know why. Yeah, so these are some of the books that I used. Not all of them. I have a stack that goes like up to here in my room. It is substantial. There's a lot of reading. But In the Land of Invented Languages is a book all about invented languages in general. So there's other ones like Esperanto is a very popular language. That one might give Elvish a run for its money and how many words have been published. There are conferences that are spoken entirely in Esperanto. So that book is focusing a lot on that and why Esperanto isn't something that can be spoken around the world, and why other languages that people have invented have failed. There's a lot of reasons why invented languages are invented and not spoken. Through the Language Class is another book that I've drawn a lot of information from. It is all about language as a whole, like spoken languages, and why they work and how they are created, which I'll get into more later. This Language, a River is a book about the history of English, which is also important. And then the Tolkien ones are obviously important because Tolkien wrote Elvish, and we want to get inside his brain a little bit. So those are that. So for my thesis, I did a lot of reading. As you can imagine, with an English major and that stack of books that I just mentioned, there's a lot of reading that happens. So most of my research was done through reading. And a basic understanding of linguistics, which I was able to do with partly with classes here at UNC, because we do have some linguistics classes, but also a lot of research on the side and reading textbooks. Yay. Once I had that confidence, I was able to take that knowledge and study the structure of English, German, and Elvish specifically. So the languages I speak, or I don't speak Elvish, but you know what I mean. To find similarities and differences. So I basically took the phoneme chart of English, phoneme chart of German, and the one for Elvish, and compared them and contrasted them and found out which sounds are consistent in all languages and which languages have special sounds. Spoiler, all of them have special sounds. It's great. But Elvish and English are pretty similar, so that was really interesting to find out. And German is there's some German sounds that we don't have in English that do actually exist in Elvish. Yeah, and then using that, we analyzed the research thing and started creating my own language. So the application of my thesis is that I will be creating my own language based on what I've learned. So we'll get into that in a minute as well. What I found is that language forms in two primary ways. There is the natural way. So when you go outside and hear a brook running around, or it's a river flowing, and you hear bees buzzing or dogs barking, like you hear that, and it's the same sound everywhere because that's how nature works. So people have taken those sounds and made those words. So that's how nature forms language. And you will notice that in different languages, the sound that a dog makes is usually pretty similar. Not the same, but similar. There's a lot of fun videos on YouTube for that. And then there's culture, because specific cultures will need specific words. There are things in America that you might not find in other countries, or things in China you won't find here. And of course, you need a word to describe that. So that's how language differs in different countries. And that's how it's formed, because you need that. And language changes based on encounter. So when you encounter a new thing, regardless of where you are, you'll need a word for it. So you can communicate that idea or that you've seen this thing to someone else. So that's another reason that words are added to languages, or that words change, because someone has encountered one thing and the other person has encountered the same thing, but they have to communicate that somehow. So then the word tends to morph. Invented languages are often unsuccessful because they are not as natural. They are very rule-based and structured, which sounds good in practice on paper, but once you try to actually speak that language and remember the words and remember the grammar rules and apply them when you're writing, it's just too much and it doesn't flow. So a lot of languages have failed because of that. Tolkien uses influences mostly from Welsh, Finnish, Old Norse, and Old English, which are all vaguely similar languages and very similar to English, which is probably another reason why Elvish is so successful, because English is so successful. So they go together. So those languages are big influences that I still have to work on studying a little bit. I know nothing about Welsh or Finnish. I just don't. So that's the next step. So the application, with all of that knowledge and that knowledge that will follow from that research and what I will be continuing to do, I will be building a language for myself. I'll be using German, English, and Elvish together, and I've already started doing it. I've created a list of basic vocabulary, so the words that we use on a daily basis, and words that elves would use on a daily basis. So like leaves and trees and battle, travel, pronouns, food, words for family. All those words that are very important are on the basic list of vocabulary. I've also created some new words using new word formation, which is the process of either combining two words from different languages, so like German and Elvish together to make a new word, or combining two words all together, just sticking them together, so compounding. Or you can put a different ending on a word, another type of new word formation. Or again, you can see how a word sounds and change the meaning of the word based on sound. That's another new word formation type. There are 5. So I don't think I went over all of them, but there are 5 of them. So I've been using a little bit of that. It's mostly combining German and Elvish together and then adding random English endings and suffixes to that. And then we've also added some just purely German and purely English to my new language, because when you are morphing languages together, you will notice that some words just don't change, they just get added. So there's words in our language too in English where we just have the French word. We just say croissant. We don't change the words, we're like early bread. It's croissant. It sounds cooler in French. We use the French. So we have some of those words in the language as well. It's just random German, but you wouldn't know that because it's a new language. Okay, so what's next? I'm not done yet. I am graduating soon, so the timeline is ramping up. I will be applying to conferences, but before I can do that and be ready, I have to continue researching how language works, going all the way back to its roots as language as a whole, not just English. I would like to know, to answer that question I talked about in the beginning, why those sounds, why those phonemes, why are those the ones that we use, and where do they come from? So I'll be doing a lot of research on that in the next few weeks. I will also continue to research Tolkien's language creation process. Like I said, I know very little about Welsh and Finnish, so I'll be doing some research on that and their language systems, which I'm sure will be similar to English, but I might be surprised. I probably will be. And I'm continuing to work on my language formation, so creating more words, making a bigger list, making my own little dictionary for that language, and working on syntax because I haven't worked on that yet. So a lot of grammar rules and how sentences work and how sentence structure functions in my new language. Yes, I think that's it though. Yeah. Thank you.
Questions for?
Yes.
So did you just discover if your process for creating your language is significant?
Yes, it definitely is. Tolkien's language creation process was very similar to a lot of invented languages. Let's look at this language. I like this part of this language. This part of this language is really cool too, so we're going to combine it. I'm kind of like picking and choosing from different languages to make a new one and using elements of that. I do know that Elvish has very similar sentence structure to English. So I'm sure he used the old English sentence structures and his own English because he spoke English to create that. And then the symbols in Elvish, which I wish I could have put up there, but Google was not my friend this morning. The little symbols are Welsh. They are Welsh influences. So speaking and choosing from different languages and then sticking it all together.
Do you have a name for your language?
Not yet.
Okay.
We're working on that. Part of the language creation thing is that we have to have a history for the language and coming up with that storyline of why this language exists. So once we have that more solidified, I'm sure I'll find a word for it. Yeah.
In graduate school, do you hope to continue studying linguistics, or are you going more like the traditional English route?
I'm glad you asked. I'm excited for grad school because I do plan on learning more about languages. The program I want to do will allow me to learn Greek, Hebrew, and Latin, so I can go into the roots of language even more. And then I hope to do a PhD in linguistics, specifically linguistics. Yeah.
So do you know when something would be considered more of a dialect versus a native language?
Yeah. I mean, I don't know this as like a firm fact, but from my knowledge, it's a dialect is more when it's before the foundation of the word is still that original language, but little accents change. So you pronounce the phoneme just slightly different, but not different enough that it's a completely different phoneme. So it's like fancy, whatever this language is. Yeah. I don't know if that made any sense, but hopefully it did.
I have a related question. So from what I understand, Elvish has multiple dialects, and I was wondering, have you just sort of squashed them all together, or did you kind of pick and choose?
I definitely pick and choose. The original Elvish, like the first edition of Elvish is called Quenya, and then the Elvish that I'm studying in particular is Sindarin, which is the next version of Quenya. So Quenya came first, and then it morphed into Sindarin. And then there's more after Sindarin, but I'm focusing on Sindarin. It is quite different than Kenya. Yeah.
Would you use your language for anything outside of your research?
I might. I do a lot of creative writing, so I might. I would love to publish a book with it, but that's a long time in the future thing if I have time after grad school. Any other questions? Andrea?
As you're building your language, you said that German and Elvish are primary ones. As you're learning more about other languages, do you see yourself incorporating more, or are you going to keep it simple to base?
I mean, I'm using 3. I'm using English as well, so that's a lot. I would love to use more, and I do find myself like wanting to because I do know multiple languages, and I'm a language nerd, so I would love to incorporate like French or Hebrew or Greek or something, but that would take me another like year to do, and I can't. So I have to keep focusing myself onto just the 3.
My question was, you said that there's the dictionaries published with Elvish in it.
Yes.
Did Tolkien do that, or are those people afterward that just went through all of his works and pulled it out and created dictionaries?
I'm pretty sure they happened after him. He was not a big fan of people like dissecting his work. You'll see that a lot in some of the books I'm reading, like his letters. People consistently sent him letters like, how does this work? Where did you get this information? He's like, just enjoy the language. Just stop. So all of this happened after he was His son helped a lot with that though. His son is very open to sending out all the information possible. So thank you, Christopher Tolkien. Yes.
What was the other inventive language?
Esperanto.
Where is that one?
That one was created by a couple of people in the 90s, the 80s or the 90s. I think it was just a spin-off of English, but very, very invented. It was not super based on any specific language, but it blew up a lot. There were conferences, books, people tried to learn it. And I think a few people were successful in like having conversations in it, but it died down.
Well, thank you, Angie. Thank you, everybody, for great presentation.
So next will be Claire, and if you are going to leave after Claire's presentation, after which is our 4 to 5 p.m. session, and you're using this for honors points, you guys that are trying to leave, hold on, did you guys sign in, or do you have this?
If you did the sign in, you would have to. I didn't. Oh, you did. Okay, well, so if you need to leave after Claire to go, I'll put one in the exact theater. Don't forget. If you already I get Claire.
If you're just coming in, come on in. We're in a great place. Do you need me to help you, or can you find it? I'll show you the easiest way. You should go to the little folder down here. Is that? Oh, here, so. All right. Cool. So our next presenter is Claire Sharp.
Hi everyone, my name is Claire. I'm a senior psychology major graduating this semester. So this is like one of my final times presenting my research. And today we'll be talking about body image and social media, specifically social media intensity and self-perceived body image. So just a little bit of background on this. I mean, social media is a big part of a lot of people's lives. Over half the world's population uses social media, and this number is increasing every year pretty much. And around 25% of time on the internet is spent on some sort of social media site. Previous research has shown a positive association between body image and psychological well-being, as well as an increase in social media use has been shown to have a negative impact on body image. Most of this research has been conducted, has looked at the amount of time spent on social media. So I was really interested in like, what are the different components or factors in regard to social media that could be impacting body image besides the amount of time. So my survey format was completely online. It took around 15 minutes for participants to complete. And there were 3 general components to this survey. The first was body image. I used a body image appreciation scale that asked questions like, I feel confident, kind of questions about that, where if you scored high on this scale, you were considered to have high body image appreciation, and low would be lower body image. Social media was a little bit more complicated, so I'll be going into that a little bit more in future slides. And then the last part was demographic questions, which ask questions such as gender, age, as well as the main criteria to be a part of the study was to be an undergraduate student at the University of Northern Colorado. So with social media, there were 3 different components to the social media part of my survey. The first was social media intensity, which was persistence, boredom, overuse, and self-expression. These 4 components were kind of have not been measured in terms of body image previously. So this is what I was mainly looking at in terms of social media and body image. The second was I also asked questions in regard to the length of time on both social media on the whole, as well as individual social media platforms. This was used in order to compare to see, is length of time a better predictor or social media intensity a better predictor of impacting body image. And then the last was just other questions that me and my advisor worked to come up with, like what would you change about social media And do you think that social media does have an impact? So these were at the very end just to gauge what are participants' opinions in regard to social media. So going more into the social media intensity scale, I use the multi-dimensional Facebook intensity scale, which I then modified to be applicable to all forms of social media. So all of the Facebook words essentially were just changed to social media, and then that worked really well. So for persistence asked a lot of questions about how much are you using it? And so like those are examples of a couple of the questions on that scale. The next factor was boredom, which was essentially, are you using social media because you are bored? The third was overuse, which is, are you using social media instead of doing other things? And then finally, self-expression, which was asking questions of if you'd use social media as a form to like express yourself. So 253 people took my survey in total. This was done through SONA, which is the participant pool for psychological students. So a lot of first-year psychology students took this survey, as well as it was offered as extra credit in a couple of other psychology courses. 238 students were used for analysis. This just meant that anyone who completed less than 60% of the survey were taken out of analysis. Everyone else was included. The average age was 20 years, as well as majority of participants identified as female. So I was really surprised by these results in that no significant correlation was found between social media intensity and body appreciation, as well as I did not find a correlation between body image and time spent on social media. And I'll be going in a little bit later to see why that might have been the case. When looking at those specific factors of social media intensity, I did find some significant results. So looking at those two, boredom and overuse, both of those were negative significant correlations, which meant that as boredom and overuse increased, so if that's kind of like the reasoning behind using social media, body image appreciation decreased. Persistence and self-expression both showed a slight positive correlation, but it was not big enough to be significant. So some limitations of this study is that it was a sample of only undergraduate college students, as well as it was mainly young adults and women who took the study. So definitely expanding populations that this research looks at would be very valuable going forward. And only 4 aspects of social media were measured. Now, I did base this off of a scale that was already published in the literature, but creating more scales that ask more questions about social media use would also be beneficial, as well as this was pandemic-based data, so that might have also had an influence on the results that I found. So this study suggests that future research should focus on specific factors of social media use, because I did find 2 factors that were significantly related to body image, as well as body image is an important factor in psychological well-being. So the research should be done as it can have an impact on quality of life, loneliness, depression, anxiety, all of these I found in the literature, as well as with social media becoming more popular. It's further research should continue to look at different components of social media. Also with future research, I think looking at different platforms would also be beneficial. I focus more on like quote unquote like picture-based platforms like Snapchat, Instagram, Pinterest, things where you'll be viewing a lot of images. I'd be interested in seeing how other sites factor into that, as well as potentially like looking at like dating apps and how that influences this research. And so that is the end of my presentation. Thank you so much. Are there any questions? Yes.
Do you think there'd be more results of the results you're looking for if you had looked at perhaps more like the types of, so Instagram, for example, the types of, I can't remember, the type of accounts people follow versus like the app itself, like perhaps the
The county column.
Yeah.
Makes the difference in the body.
That's something that I really want to look at because I definitely think that it might have an impact, and that if you're looking at specific accounts that might have like a bigger impact. So definitely like in the future, that's something that I really want to look into. Yes.
On the variable of overuse that you said did show the significance to be negatively correlated with the body image, what was that coming from the reported number of hours, and how did you determine that something was overused or not overused? How did you kind of dig into that?
Let me go back a couple slides.
Yeah.
So overuse was measured with questions kind of like this one right here. So there was 4 questions in total. These are just 2 of them, but it asked questions like, I spent time on social media at the expense of my obligations, and it happens that I use social media instead of sleeping. So that was asked based off of self-reports of if you viewed yourself using social media instead of doing like other things. Are you doing it instead of eating, sleeping, kind of things like that?
Okay, so it wasn't based on the number of hours. It was really the person's self-identifying that they think they're overusing it because they're doing it in kind of a negative use. That's interesting.
Yes, yeah. So length of time I did look at as well, but it was measured separately. And so this was the focus of my research, and then length of time was just used as that is like what's predominantly used in literature on social media and body image. So I was just curious to see if this was a bigger impact than the length of time.
Did you come up with any results based on length of time? Or-
Yeah, there wasn't any like correlation between time and social media. And there also wasn't like between social media intensity on the whole and body appreciation. It was only when I broke it down into those 4 components that that significant correlation was found. Yes.
For your open-ended questions, in your poll, did you find like a common answer that people wanted to change of social media?
I haven't fully looked into that yet, in that those questions had some multiple choice, but then there was an other box that a lot of people filled in. So I still need to go through and like kind of thematically go through it and see. But overall, people did like agree that did believe that social media was impacting their body image.
Did you find like a connection between the use of a certain like social media platform and like probably that they're using to report like TikTok, like more likely how you use Instagram?
That's also something that I'm doing. Those are all like the analysis that I'm working on just to kind of finish it up based off my advisor's feedback on like my research so far. So I will have that answer by the fall symposium. Yes.
Do you think if you looked at like middle schoolers who have the opportunity to be significantly less exposed to social media, you would get different results?
I believe so. In the literature, there is a difference between different age groups. Most literature has focused on young adults in the past, but when looking at young adults compared to younger populations and older populations, there is a difference. I didn't look at participants under 18 just mainly because of time constraints and getting IRB approval, but I do believe based off of literature that's been published in the past that there would be a difference.
So thinking about your questions for boredom and overuse, right? Obviously those are kind of related, and it seems like they also maybe correlate with an absence of a feeling of accomplishment or self-care, right? Because part of the question is like, am I getting enough sleep, or am I doing the things I'm supposed to be doing? So I'm just curious, have you thought about kind of other factors of well-being, things like a sense of accomplishment, or people who are engaging in active self-care? Or are you kind of trying to separate that out because that's a little too complicated? But I guess my question is, have you thought about the relationship between these other factors of like positive body image and social media?
Yes, that was actually originally what I wanted to do, is I wanted to look at social media and body image, and then look at like how psychological well-being and factors within that can mediate the relationship between the two. And through talking with my advisor, I kind of transitioned my project into more basing on this. I am applying to graduate school currently though, to work more in, not necessarily this topic exactly, but looking more into social media and technology and influences of that across well-being.
All right, thank you so much, Claire.
Okay, so that's the end of our 4 to 5 p.m. session. So again, for those that were Honors 101, students, if you're only going to be here for the first session, then for our folks to log and run to the back door, or if you signed in on the pending, right, some help even after that. But otherwise, we're going to launch right into our 5 p.m. session. So, Katie, are you next up? Let's go ahead and we'll get your presentation up. You hit escape. You know these. No, you just did that one. Oh, there we go. Yeah. Did you get yours? Get logged in or You're welcome. Thank you. And before we launch, we got a minute, so we'll give the 60 seconds to wait to see if we have any 5 p.m.ers coming in. Most people really sign up just to be here the whole time, which is just great. Couldn't stay. But the one thing I did mention at the beginning of the first session was, if you are here and you're in Honors Interdisciplinary Program, and I think there's a few of you here that are still in Honors Interdisciplinary Program. If this is sounding like something you want to kind of launch into, and you realize you as well can design your own original research project and dive into that, we are doing an information session. I put a flyer towards the front door, Andrea. I think it's November 9th, correct? Like lunchtime on November 9th, we're going to do a little info session on what that means to transition into upper division honors. But you can also ask questions if anybody's presenting now, they've got a good sense of what upper division honors is all about. And so you can transition into that starting in spring. And Corinne will be teaching Honors 351 again. Do you want to say 2 words about it? I'm going to put you on the spot, but anything you want to say about getting started in honors? I mean, upper division honors?
Solid question. I mean, hopefully there are 351 students in here, so I can't oversell it, but hopefully 351 is kind of exciting in a nice way of getting into research and kind of thinking about working as a scholar rather than just as a student. So that's kind of what we focus on in there, sort of getting you ready to design your own research projects that will eventually result in the kinds of projects you're seeing today.
Yeah, and it is an iterative process. I don't think that anybody that you've seen present so far, that when they first started on that first month of Honors 351 in the first semester, like, this is exactly what I'm doing. All of these students, this developed over time, and depending on how much time they had, kind of, we find that the longer time you have, the more it kind of develops more slowly, and then students on shorter time frames are like, okay, I gotta just like kind of pick this and dive in. But if you have 3 to 4 semesters, that's a great time frame for getting started in honors. And I think, Claire, you started in 2nd semester of your sophomore year, right?
Yes.
Yeah, so we do have students that start 2nd semester of their sophomore year in that phase. So please come find us, talk to the current students that are in here that are doing this if you're interested. I think it's the one of the most worthwhile things you can do as an undergraduate is doing a project. So, all right, with that, Katie, go ahead and tell us what you've been doing.
All right. Hi, I'm Catherine or Katie Johnson, and I am looking at misinformation of mental health on social media and how it affects those who read it or really kind of see it. It's a little interchangeable that way. So just a little bit of like a background definition session real quick, just so everyone's on the same page. Misinformation, I'm looking specifically at a claim of fact that is false due to lack of evidence. So if you're seeing something where it's not fully backed by research or isn't something that's kind of like everyone knows it, then it would be considered misinformation. Social media, I'm sure everyone knows exactly what that is. We're on it all the time, every day. That would be like the websites and applications like TikTok, Snapchat, Instagram, Facebook, any of that kind of stuff. And then mental health is another thing that everyone kind of knows, so a person's condition with regard to their psychological and emotional well-being. So why am I looking at this research specifically? So I found when I was in Honors 351, I was looking at this little section of research that no one has done anything on. Found this nice little niche where I found no other research studies and there was no like literature on this specific area. There's tons of stuff on social media and misinformation, social media on mental health, and misinformation on mental health, but not all three of them put together. So I was like, that's what I'm doing. I'm going right there. So and that brought me to the research question of how does the misinformation of specifically mental health and how that is viewed on social media affect like the stereotypes, the attitudes, and stigmas believed by those who have viewed the misinformation. So I'm looking specifically into how the misinformation that is viewed on social media affects how other people see mental health. So my methodology, I'm doing specifically a quantitative data analysis. So I'm looking at the numbers, and I did a survey through Qualtrics where they do a little pre-test with some demographics of like if you're male, female, what age you are, if you're like freshman, sophomore, junior, upper division, all of that kind of stuff. And then I split everyone between two different videos. They were randomly assigned. So I have two TikTok videos that I made with both of them having some misinformation and some what I call true information, where they directly correlate. So one video will have one question or one statement that is directly correlated in the other video, where one is a false statement and the other is a true statement. And then afterwards, I have them doing a post-test, which is the exact same questions as the pre-test. And I'm mainly looking at those differences in the numbers of those two tests. And then I give them a disclosure statement saying, hey, you saw some misinformation. Don't take everything in that video as fact. Here's the true information, because I don't want people leaving being like, oh, I learned something new today. I am specifically looking at TikTok videos and depression, since a lot of people are on TikTok nowadays, and a lot of, at least the media that I see on TikTok, is about mental health and specifically depression, of being like, if you're sad for more than 2 weeks, you have depression, and a lot of people tend to take that as fact. For my participants and results, I wanted at least 50 participants, and I was getting them through SONA. I currently have 64, so I've reached my goal, which is very exciting. And I'm doing data analysis through Excel or SPSS, which is what I am currently doing for the rest of the semester. So getting into some of the preliminary results that I have, we have found that, we split it up between our true facts and our false facts. So for each of the videos, there's like what I call the true facts, and then there's what I call the false facts, where some of them are completely true and factual. I have found these through research and through the different resources that I've looked at. And some of them are false facts where I like changed one little word, or I have made it just enough different that it could be taken as true, but it is false. So we have found, surprisingly, that there has been an increase from the pre-test to the post-test for the true facts. I believe most of it is because most of my study participants are psychology students. And then there's been a decrease from the false facts from the pre-test to the post-test. And that is for both of the videos and just the regular averages. And then going more specifically into the different videos, there were two questions for video 1, question 30 and question 34, that had a significant difference. Question 30 is, to be diagnosed with depression, the symptoms must be at least 2 weeks, or must last at least 2 weeks. And that one increased by 0.59. So if they said like a 3 for their pre-test, then they said around a 4 for their post-test. And then question 34 was, if someone in your family has depression, you're almost guaranteed to have depression, which is counted as a false fact, because you're not guaranteed to have depression. And that one increased by 1.45, which I found very interesting. But the video that they watched for video 1 actually said that if you had a family member that had depression, you were guaranteed. So that was really cool to see that that video kind of affected people. And then for video 2, there were 6 different questions that had major differences, which was really awesome to see. And 2 of those questions were the exact same as video 1 and had somewhat of the similar increases. But I did find that all of the like factually false questions that like within research are not true had all decreased by over about 1. So that would be question like 64, 65, question 57, and some of those other ones where they were factually false, but they decreased. And that didn't change depending on if the video told them if it was true or false. So that was very interesting research for me to see. So the rest of my timeline for this semester is I'm finishing collecting data until the end of October. I am planning on finishing analyzing all of that data by the end of November, and then finalizing my paper by December 1st for the deadline. And I graduate come December 10th, so I'm really in like that final little run. I started all of my data collection and analysis at the very beginning of the semester, so it's been a very short timeline. I wish I had a lot more time to be able to do everything, but unfortunately I did not. So yeah, any questions?
Thank you. Question for Katie.
Yes.
Do you think that this misinformation is malicious for the intent to just gain fans and internet popularity? Or do you think it's due to the fact that now anybody can just Google or look something up, and what comes up on the internet isn't necessarily true, but it seems true, so they just put that in the video?
I think it's a little bit of a mix of both. A lot of people do put out misinformation just because they don't know that it's misinformation. Like we have a lot of people that will look up like the DSM diagnostic criteria for depression, and because they're not a professional, they can't fully understand it and like put it out there for people to fully understand. So there's definitely parts of that, but there are also some people who very much put stuff on the internet just to like become TikTok famous and are just saying things to say things. Yes.
So when you were, when you asked like a question, and you had to watch video, and then you answered like a similar question post-test, and there was like, so if there is an increase of 0.5 or an increase of 1.4, what does that mean? I'm struggling to understand what the numbers are saying.
So my I did it based on a Likert scale, so they can only go 1 to 7, and there's no like in betweens for that. All of these numbers that I showed up here were all averages. Go back to them. Yeah, so all of those numbers are averages of all of the data. I believe it was like 47 participants that I looked at for each video that were like condensed down. So those are all averages of like between all of the numbers that they put of like the 1 to 7, that's like the average number. And then these were averaged down even more for like the difference between the pre-test and the post-test. So what I found is a lot of people, if they decrease from the pre-test to the post-test, what they're looking at is their belief from going into the survey has decreased of how true that they believe that test. So like one of the questions that I have is like, to be diagnosed with depression, the symptoms must last at least 2 weeks at a Likert scale of like 1 being that's not true whatsoever, 7 being that is very true. So if they started out as a 7 and decrease it to like a 5, then their belief of that question decreased after watching the video. Sorry, that was a very wordy explanation.
You still have one.
Any other questions? Yes.
Do you think there'd be a difference if you didn't have psych majors entering this?
I believe there would be, just because everyone that kind of goes into psychology knows like the basic stuff about depression and everything. So if I was to put this out to the general population, I feel like there'd be more of a difference, especially because social media is for the general population. So it would be a little bit more accurate to what the answers would be like, because not everyone knows that in order to be diagnosed with depression, the symptoms have to last at least 2 weeks. Or like the opposite for that question was, like if you're feeling sad all day, every day, then you have depression. Oh, yes.
That disclosure at the end that you give them is really important ethical stuff, and I'm just curious, was that your idea, your advisors, did IRB make you do that?
That was my idea. I find, because I see a lot of TikTok videos that are like, if you have ADHD, this is what it looks like. Or like if you cry yourself to sleep every night for like a week or whatever, then you have depression. And I'm very much like, that's wrong. That's not how it actually is. So I wanted people leaving my study to fully know the true facts and be able to be like, oh, what I viewed was incorrect. Maybe I should fact check myself when I see something like that similar in like TikTok videos or on Instagram or anything.
Just to clarify then, so you really did find quite a bit of significance in that people were influenced by the TikTok videos, right? That's kind of
Yes.
your ultimate take
Yes.
on this, is that they were influenced by the TikTok videos.
Yeah.
With that information giving them then things that they believe that are not true, right?
Yeah, like for video 2 with those like big numbers, that like 1, that means that they move their like little marker for the Likert scale down from like 6 to 5 or even like 4 to 3. And we found with a lot of those videos for like what I call like the factually false facts, so like the ones that are completely false altogether, those people tended to, or my survey participants tended to put them more as like a neutral, as like a 4 or a 5, where like they didn't quite fully believe that it was completely true, but they didn't quite fully believe that it was completely false.
Right.
So it was very interesting to see that, because specifically for video 2, at those like questions, some of them were, or most of them were told as like true in the video. So it was interesting to see that they decreased that number, even though they were told as true in the video. And like some of them that were told as false in the video did also still decrease. So it was very interesting to like see those differences. And I'm hoping that those kind of increase more with the more participants that I get.
Right. Other questions for Keith? Okay, thank you. Richard, are you ready?
Yeah.
Because Kayla just walked in. Do you want to take a break and Richard can go first? Okay, why don't we do that? We'll keep him in. You both are running a little bit late and we'll do that in advance. So it's just like a race. If you got it.
NAFLD, right?
Yeah. Oh,
Okay, cool.
thank you, Lauren.
Right, you should be good. All right, so next up is Bridger Menlove.
Howdy, y'all. So, mouthful, my project is therapeutic effects of cannabidiol on innate immune cells in non-alcoholic fatty liver disease. I'm going to run you guys through the basics. What is NAFLD? NAFLD is a disease that is only characterized by one thing, 5% fat accumulation within your liver. This disease, since it's with fat accumulation, has to do with things like diabetes, obesity, metabolic syndrome. So it's an not an eating disorder, an eating disease. And so how it happens is you get that beautiful, greasy, cheesy hamburger and you eat it and your body is built to preserve all of that extra nutrients, the proteins, the fats, maybe even some sugars from your milkshake. And this gets sent, all of it that is not actively being used gets sent off to the liver for packaging. This is one of the liver's main roles, is taking all of the different things you consume, throwing it into these nice little packages we call triglycerides, and either storing it itself or sending it off to fat cells. And so the liver plays a really key role in what we eat and what happens to it after it's been eaten. I will note that a really important part of this disease. And also, it's not just the diet, it's how the diet affects your micro biome in your, especially your large intestine, and that will also play into this disease. And what occurs is you develop a fatty liver, which we call NAFLD. You would be surprised, 21% estimated of the United States adult population has this disease. And you'd say, well, one, why is it estimated? And two, why have I never heard of this? And that is because there are actually no negative symptoms of non-alcoholic fatty liver disease. And the problem with that, what makes it really scary, is then people are running around for years with this disease, and somewhere around 20% of them are going to develop NASH, which is where an inflammatory response occurs, and you start to see dysfunction, you start to see cell death, and eventually your liver literally turns into a bunch of scar tissue. Scar tissue is no longer viable, your liver quits working, and the only current treatment for end-stage NASH is liver transplants. And so if you reach that point, late-stage cirrhosis, you are very, very likely to develop liver cancer, which is also only treatable by liver transplants. And so this puts us in a really scary position because this is the fastest growing liver disease worldwide. And this is compared to your communicable diseases like HIV. These are considered liver diseases. And so even though this is a food disease, it is sweeping across the world and is untreatable. And so currently, and so what we know is this, sorry, right here, this switch from just a fatty liver to an inflamed liver. This is where you start to see all the negative symptoms, pain before and when you start initiating eating, pain at night, scarring, and all of the smaller things, oxidative stress, apoptosis, which is just essentially your liver slowly dying. And so what this is it's a whole bunch of immune cells running around, talking to each other. This graphic illustrates really well that all of these immune cells are acting upon the liver cells themselves. These are hepatic stellate cells, causing them to create the scarring that defines NASH. What it doesn't show is that there'd be too many arrows. Let's call a bunch of this activates this, which then activates this, which then activates this. And so to keep it simple, your first immune cell is going to be this, Cooper cells. They are resident to the liver, and they are your light switch. When they turn on, they will bring in B cells, which will bring in T cells, which will bring in CD8 plus T cells, which will bring in neutrophils. And this whole crazy mess that we call NASH occurs. And so it all starts when these cells start to realize, hey, there's a problem. Cells are dying, cells are being stressed, and we need to make a response. We are the first line of defense. And so what we've been looking at is there's a really odd study where cannabis users were found to have lower incidence rates of this disease than the general population. And so because of that, a whole slew of researchers went into CBD. And what's happening? It's likely these non-psychoactive therapeutic agents, CBD, CBG, CBG, and others. And so CBD has been shown in this disease as well as others to decrease inflammation. The thing we're looking at is CBG, another non-psychoactive component of cannabis. And what makes this interesting is it actually has better agonistic effects. It's acting in the same ways in the heart, and it's a harder hitter than CBD. But I would say the reason why CBD is being researched more is it's a little bit easier to extract, and it comes in greater quantities. but it doesn't mean that that is going to be the best therapeutic treatment for your disease. And so what we've been doing in my lab is looking at CBG effects on the disease. We use a mouse model, so we induce the disease by feeding them not hamburgers, but a high-fat meal. And they develop the disease, and then we treat them with oral CBG, euthanize them all, and then we look at their molecular, the molecular level of their liver. And so there's a whole lot of projects going on with this. We've seen it messes with the structure of the fat in the liver. It will reduce, this is the related to the fibrosis, the level of the level of fibrosis occurring. It reduces organ size, which is something when you're inflamed, things get bigger, things get swollen. And so mine is on what's happening with the immune cells, because we know that this CBG is acting on, we saw PPAR gamma, CB2, and A2 adrenergic. And so it's activating these immune cells. We know they have these receptors, and so we know that there's some sort of light switches going on. So the question is, what's it doing? And so I'm focusing on the starting point. Macrophages turn it on. The next one is B cells. These guys are your innate immune system. They're the quickest to respond. They are the most prominent in this flip from a fatty liver to an inflamed liver. And so what we've been doing with these mice is we take their livers, we section them out, and we do a whole bunch of things, looking at proteins, looking at RNA, and trying to determine what is changing within these very specific cell types, and within these cell types, what seems to be happening that's decreasing these inflammatory things we're seeing on a gross scale. And so I looked at splenic B cells, which is where B cells are usually housed in your spleen, and it's literally a balloon, and it'll squeeze the balloon and shoot them off wherever they need to go. Interestingly, there is no change in B cell populations. This is where they're going to proliferate. This is where you're going to get new ones. And so it's interesting that they don't seem to be the starting point. They're not activated first. They seem to be coming along later because there is an increase in hepatic or in the liver B cell populations. And so something has to be bringing these guys in. They're not activating themselves in the spleen and they're being recruited from outside, and they're not being recruited specifically from the spleen, because they will, they will also be outside of the spleen, flowing through the blood. They're one of our innate cells that are ready to attack foreign invaders. And so something is bringing them in and increasing the levels of these kind of proteins, which is just a protein cell for B cells. And we see that it's only significantly increased in our CBG diet, which is weird. You would expect, like, if there's decreased inflammation, then there should be less immune cells. But what we find is that weird macrophages. So they have an increase in TGF-beta and CCL2. These are recruitment genes. This is an RNA analysis, and we see an upregulation in these recruitment genes, which say, well, what the heck? How is it that they're recruiting, but they're decreasing inflammation? And this has to do with the activation state of the Cooper cells. They have two light switches, essentially. One of them says, hey, everybody come here. Let's throw a pro-inflammatory party. Let's go crazy. Let's cause fibrosis, steatosis, wreak havoc. And the other one is anti-inflammatory. Hey, everyone, let's come here. Let's remodel the fat. Let's get rid and clean up all the dead cells, and let's reduce the inflammation, which is weird, because you think more immune cells, more inflammation, but it's more about what is happening within the immune cells as they come in. And so these macrophages, these two specifically, are much more correlated with our type 2, the anti-inflammatory state of macrophages. And so we believe, this is where, oh, it's a never-ending project, where we know based off of the lipid remodeling and the changes within these RNA that macrophages are polarizing towards M2, anti-inflammatory state. But then the forever questions are, well, why? Well, what happens next? Well, what about the pro-inflammatory cytokines? What are they doing? They should be decreased. And so that's my next one. I want to really make sure that we're not only seeing an increase in anti-inflammatory, but we're seeing a decrease in the pro-inflammatory cytokines that these macrophages are also known to release. And so we'll see what happens. I would like to acknowledge the OUR. They have funded a lot of this work. I've been very grateful for them, as well as my professor and one of my grad students. I took a lot of his tissue, and so I'm very grateful for him. So, any questions? Please.
So I noticed if you go back to the liver slide where it has like a transition about what was it?
NAFLD to NASH.
Apple to NASH, yeah. So you said that a patient, for example, would experience pain prior to eating. So would that be like an anticipatory response to knowing that you're going to eat?
Correct.
Okay.
It is also more likely during certain times of the day because circadian rhythm, your liver preps itself, especially in the morning, says, I need to be ready, regulates a ton of genes, and then for whatever reason, down regulates them at night, destroys them all, and goes through the same process. So you'll see right before eating, as you get that response, the brain liver axis, and as well just during certain times of the day. Increases in pain because you activate the cells are, it's like guy with a broken leg. You tell him to go run, it's going to hurt more when he's running. So, yeah.
So if this turned out to be successful, would you recommend then that people like be taking CBG to prevent like NAFLD and NASH to cirrhosis if it's asymptomatic?
So something we need to be very careful of is I'm looking right here, but this disease can last for years and it changes. NASH is much more affected by some of these guys. T1 cells, T8 cells, natural killer cells. And so what we haven't looked at is, well, how's CBG acting on those guys? So I would definitely not suggest with where we've reached so far, this is not something that we know helps in late stage disease. We know that it's acting in a good way on early stage disease. And we also, and so I would suggest there, but also it has not been, it hasn't been fully studied what it's doing elsewhere in your body, or as well without the disease. And so something we're doing right now is we're going back and we're just giving my CBG to see, is there anywhere that we're just seeing adverse effects? And so, especially within a healthy liver, you try to give someone this and they're preventative, and it may actually do them harm because it's over activating certain cell types that don't need to be activated. And so that's the kicker is we only know one specific point in a very long disease. Does that answer your question?
Yeah, no, that was awesome. Thank you.
Yeah. Yes.
We're using it with that TDM data as opposed to the 5
I will look at 1,000 of them because it gives you a better picture. But TGF beta is very strongly correlated in literature with M2 type macrophages, as well as CCL2. Less highly correlated, but we would expect to see increases in this. But I do agree that is fun fact in our lab what we're doing. IL-10 pro-inflammatory, IL-10 anti-inflammatory, and IL-12 pro-inflammatory, to hopefully really narrow down the decrease in our pro and an increase across the board in our anti-inflammatory. Any other thoughts? Yeah, okay. Anyone else? Please.
One more question. So with the high fat diet of the mice, so how does that compare to the actual fats that are in, I guess, human diet?
So the liver processes everything in the same way. All fats turn into triglycerides, and we use the same set of enzymes as them. So you are looking, for the most part, you're looking at the same genes in the same cells, and so it's very easy to say if we're seeing an increase in these genes specific to lipid remodeling in a mouse, we're very likely to see it in humans. One thing mouse model sucks at is male-female differences. We see several male-female differences in the mouse model that do not correlate to humans in all studies of this disease, not just my my project, but when people are really looking into it, we see mice run into certain problems that we don't. We run into certain problems they don't, including just flips in gene regulation. So we try.
That's pretty cool.
Mhm. Please.
Could I see the QR code for the references, please?
Heck yeah.
Librarian in the room. Thank you.
Nice.
Other questions for Bridget? All right, awesome.
So our last presenter today is Kayla, and did you send me? You did not.
I did.
Oh, you did.
I think it was a little late though, but I have a secret. Let me just post that. Hold on. Yeah, there you are. Yes. Looks like you can. Yeah, I'll just slide through.
Okay.
We'll work that.
All right, cool.
Yeah.
Awesome.
Thank you.
Thanks. All right, our last presenter today is Kayla Garza.
All right, so I have a PDF, so I'm just going to be sliding through slides, but my name is Kayla Garza, and I'm studying the circadian rhythm in vitro just meaning outside of the body. And I was mentored by Dr. James Hoyen and Luke Krinsky, who's now off to med school. And so we can begin to ask ourselves, well, what is the circadian rhythm? And so we can think of it as our body's clock. And so it's our body's way of keeping time for us. And so we're all familiar with the Earth revolving around the sun, and upon revolution it's also rotating, and so we get these 24-hour day-night cycles that we're really familiar with, and it's these 24 hours that cause oscillations in our body or different changes in this 24-hour period. And so we get changes in our blood pressure or temperatures, the way we regulate our temperature, and just the hormones that are secreted. So we're familiar with melatonin, and so that's really high in the evening because we're anticipating nighttime. And so why I was studying the circadian rhythm is because I wanted to look at the clock genes. And so I had chosen the ones in red. So I have period 1 through 3, cryptochrome 1 and 2, and BMAL 1 in clock. And so this whole circadian rhythm, so these changes in these genes that are keeping time for us, so they're actually in the hypothalamus of the brain. And that's where we have a master clock, which is termed the suprachiasmatic nucleus. You can just think of it as the master clock. And so we take in all of these external cues in our environment. So we have the sun, and that's one of our timekeepers for us. So it's a light beaver. And we even anticipate eating, like Bridger had mentioned. And so all of these things are related to our circadian rhythm. And so just to give you a broad overview, so we actually have clock genes throughout our body. So we can think of them as little clocks. And so they're in our periphery, so you can find them in the blood, the immune system, so in the immune cells, T cells, And in your muscle, I would be surprised if you can tell me somewhere where you can't find these peripheral clocks. And so they're actually all synchronized to this master clock in our brain that I had mentioned. And so there's a really complex pathway, but we can begin to ask ourselves, well, what happens when this synchronicity of the master clock to our peripheral clocks, well, what happens when that's broken? So what happens when our liver clocks fall out of sync with the master clock? And so we can begin to look at the future implications or what actually happens. And so a 2007 study had actually rendered night shift work to be equivalent to a human carcinogen. And you might ask, why is that? And so there's night shift workers that are exposed to this ambient light or this artificial light, man-made, at weird hours in the night, and they're obligated to do so. And so it's inevitable. And so it's been proposed that disrupting the circadian rhythm and the synchronicity of these clocks in the body, that it actually might be driving cancer or tumor formation, tumorigenesis. And so you think about healthcare workers and first responders are all subjected to this stress. And it's just going against everything that's evolutionarily ingrained in you from your cells, because you're anticipating going to bed at a certain time and waking up at a certain time. And you know when it's going to be day and night, but working these shifts disrupts that whole system. And so, like I said, with our extrinsic cues like our sun, so when we take in this light, it can actually drive tumorigenesis. And there was a study done on women with light perception and without light perception. And they had actually found that there were significantly higher rates of breast cancer in those that could see. And so they're able to take in these offset times of day where they're exposed to light or not exposed to light. And so you begin to ask, well, is this a coincidence that these women that can see, they have higher rates of breast cancer and they're subjected to this artificial light? And the same thing was happening with a cohort of women in Denmark, and they were actually working night shift work. And so they had over 10,000 mutations cumulatively, and they were all in clock genes. And so it really comes down to the fact that it's not a coincidence anymore. And they've actually found this not only in night shift workers, but pilots, because they're flying in the night and they're flying in the day when they should be sleeping. And so what's lacking is an application on triple negative breast cancer, because these studies throw around breast cancer as this really broad term. And so there's no specialization in a particular type. And so triple negative breast cancer, so it's really aggressive, and that's what separates it from almost every other breast cancer. And so we're missing the estrogen receptors, the progesterone receptors, and the human epidermal growth factor type 2. And so all of these targets that are used in treatment plans, they're missing, and that's what makes it really hard to target. And you'll see on the morphology on the bottom, so we have a really uniform tissue on the left, but it slowly gets more, you have more aggregates of cells, so it's non-uniform. And so my first question started out really base level. So are these cells, when they're disconnected from the nervous system or the cardiovascular system, when you're no longer regulating your temperature, when they're outside of the body, are they expressing these clock genes, and do they need to? And my second question was, well, if they are present, are they showing a circadian rhythm? Are they oscillating and changing throughout the day as they should be? And the third question was really the big one. And so are these cells and are these genes responsive to an external source of red light? And so I chose red light because red light's actually been shown to alter mitochondrial function. And so if you've taken a bio class, you know it's the powerhouse of the cell. And so what red light does is it actually stimulates the mitochondria and this fluorescently activated enzyme. It's cytochrome oxidase C, or C oxidase. And it actually increases the levels of ATP, and that makes the cell more efficient, and hence it could have anti-cancer properties, but that's still up for debate. And so the way I had set up my study, so I had 3 groups. So I had one group of cells exposed to ambient light, regular classroom lighting that a night shift worker might be exposed to. And I also had a group exposed to red light. So I actually had a light in the incubator exposing that light onto the cells. And then I had a control group, which was in complete darkness. So I had covered them in tinfoil as a means of excluding the light. And the way I carried this out, so I had cultured the cells for about 2 weeks, and then as a means of kind of resetting the rhythm, because I didn't know what they were going to do gene-wise. So I had covered them all in tinfoil, as you'll see in the middle, just to have kind of like a baseline for them. And I ended up harvesting the cells at different time intervals. So at hour 0, so I came into lab at midnight, and then I came in 6 hours later at hour 6, 12, 18, and 24. And so that was a 24-hour experiment. And each time I harvested the cells, I froze them down in trizol. And what trizol is it's a really nice buffer for RNA because it's super unstable. And so that's what you'll see in the pink. And then just for non-bio majors, so what's in these pink samples right here is RNA. And so we have genetic material and non-genetic material. And what we want is the cDNA, so the single-stranded with just genetic material. And so with RNA isolation, we're basically taking that RNA out of the pink sample, and there's really just everything that was in the cell in that sample. So you have the membrane, which is broken apart, you have all the parts of the cell there, but we really just want the RNA. And so we had gotten these nice curves that you'll see in that graph. And if we had gotten a ratio of 2 for our concentration, we knew it was high quality, and it worked out nicely because all the samples were of a ratio of 2. And so with cDNA synthesis, we essentially used the kit, and once we got the cDNA, we had ran it through this machine here, so qPCR, and with each of our genes, we got a really nice bell curve, and if it was solid, like you'll see right there, that's how we knew we had our right gene, and anything outside of that was noise. So, sorry, that's a lot of science jargon, but this is the fun part. Okay, so with our oscillations with ambient light, so when these cells were exposed to classroom lighting, you can see that the rhythm is kind of out of whack. They're kind of doing their own thing, and you even see like period 3 jump up on its own around 24 hours. And so we can kind of pull from that there's not a proper circadian rhythm there, because they should be following a synchronous line like we see in red light, which is really interesting. And so we see BMAL1 and clock following a similar trajectory, so in dark blue and green. They actually work together in the body, and so that actually supports the fact that the red light is enhancing a more proper and a healthy circadian rhythm that's often lost in cancer cells. And so we were able to answer yes, that these cells are rhythmic in vitro when exposed to an external source of red light. And then this one was solely based on the control group. So these ones were in complete darkness. And this is exposed to a gene that is always on, so it's constitutive. And you'll see that they are rhythmic, but it's really not a clear and fixed rhythm that's healthy, as we had seen with the red light, where we see these clear oscillations throughout the day. And so we were really able to answer that, yes, these genes are expressed in vitro. And 2, they do express rhythmicity, even if it's not proper. And 3, that red light can induce a more proper rhythm. And then for limitations, so I had actually lost my whole second trial because the PCR failed on me, which is often what happens in lab. And I had lost hour 6, and so my data is showing everything except that hour, and so I'd have to rerun a lot of samples and a time crunch. So, sorry. Yeah, this was done in 1 semester, so it was really expedited. So I had 1 semester to master all the protocols and then carry this out. So I did it. It was just on a really tight time crunch. And I would want to run this in mice models just because the in vivo model is just so much more dynamic and different than cells in a dish. And so I think that would be really meaningful to pull from. And these are my references. I didn't have a fancy QR code like Bridger, but and this is my contact information. So thank you to all my mentors in the OUR grant. They funded a big part of this research as well. So thank you guys.
Questions for Kayla?
So I have a question about the red light.
Mhm.
So the red light has proved to be helpful, right?
Yes.
So how does this would affect your eyesight? Because I know your article full of like photoreceptors and photobots, and
Mhm.
your cones are especially exposed to certain colors that can really mess up the rods and cones and like how your color vision.
Mhm.
So exposing like night workers to like red light,
Mhm.
how would this affect their eyesight?
Their eyesight, that's a great question. I think for one, it would depend on the exact intensity of that light, but then the exposure longevity that they have, so how long they're exposed. So these cells were exposed to like an hour of red light. So it really didn't take much to induce this proper rhythm. But that would be, I'm not sure about the eyesight because I don't think it would be damaging, but I do know that it would influence your genes. Because with, for example, the night shift workers, oftentimes, are you a biology major?
No, I'm in.
No? Okay, yeah, that's same thing. So with night shift work, you can actually get these mutations that add on chemical structures. And with this red light, it can actually take those off that are inducing those mutations and often driving tumorigenesis. And so it'd be interesting to compare that to people with pathologies, with cancer and without. But with eyesight, that's not what I focused on. So I'm not sure, but I don't know if that gives you something to pull off of. Oh.
So did you look specifically at people who like randomly do night shift work, or did you look at people who mainly like always do night shift work?
Good question. So it was actually long term. So it was like over decades rather than like if you pull like a night shift on the weekend. Like I don't think that's going to cause you to get cancer. Like I don't want to freak anyone out, but yeah, it's definitely a long-term thing. Yeah.
Then I have a follow-up to that. If you were to do like a few nights where you only work night and then like you flip-flop for like the next couple of days and like we're away during the day, do you think that could affect it and like how long it takes for it to happen, or do you think it would still kind of happen?
I think it might have a really similar impact just because you're conditioning your body to work, for example, graveyard chart, like shift. So you're working the back end of the shift and then you're flipping to the mornings. And so you're really throwing off your circadian rhythm because it thinks it's adapting one way and then you switch it right away. And so you're still getting that asynchrony. And so your genes, everything, they're picking up on that. So yeah, I think it would have a similar impact. Yeah.
You mentioned light as a major factor that disrupts the circadian rhythm. Might there be other stimuli that cause the role, like sound or touch? Might that be an option as well?
Okay. So in terms of the circadian rhythm?
Yeah, like for example, like night shift workers used to reach while they're staying awake for that. The sound that also seems to be impacted. Are those other sensory possibilities?
Right. I think those would definitely play a role because one of the genes that I mentioned at the beginning, let's see. No, there's one. Hold on. It's BMAL1. Okay. So that's brain and muscle, and it's like ARNT, like one protein, but that's incorporating clocks in your muscle and your brain. And so when you're moving and you're interacting, you're working at night, you know that your clock genes are picking up on this, as are other cells. And I guess they kind of tell, so your nervous system and these sensors in your muscle, they know that you're doing things. And so I think that would impact the oscillations that you see in these gene expressions. And so I think, yeah, whatever you're doing, like movement wise, locomotion, what you're listening to, those all play a role in this complex pathway. And this gets into it a little bit with the molecular circuitry, but that's just really surface level. But I do think that would have big implications as well. That's a good question. Did I answer your question?
Oh, yeah.
Yeah, I don't know those specifics, but I do know that it has an impact. Yeah.
Was there a reason why you didn't do the controller straight up sunlight? Was there a reason why you didn't dark as the components?
I think I was mostly inspired by night shift work, and so you're really not exposed to the sun or the natural light, but that would have been a really interesting group to compare to, because that's a totally different source of light. And my dad's a night shift worker, so I have kind of a internal want to do this as well. So yeah.
All right, are there any other questions? All right, cool, thank you, Kayla. So before we excuse everybody and have, especially those of you that didn't sign in on our sign-in sheet, do our checkout, I wanted to have several of the students that presented this evening are graduating this semester. And since we don't do a big
Honors banquet in fall, I want to have you guys stand up and come up here, which is should be Kayla and Claire and Katie. And anyone else, Jeff, you're not. Okay, so these three, I want to give them a big applause for all of the work they've done. And congratulations for graduating. You know, there's still a little bit of time left, but this presentation always feels like it's a little bit of a culmination. So thank you for coming and presenting today. So now I have these things. I'm going to just kind of pass them around. And there's one at the back door, but I don't know if Corinne, if you want to hold your 351 students or say anything else to them before they're excused, but other than that.
Sign in. Also, wherever the sign-in sheet ended up, please make sure it gets to me. Oh, yay. Okay.
No, you can just do the other one. This one gets some feedback though, so if you want to get feedback on that. All right, yeah, you guys can sit down. Are there any other questions? Anything? Yeah, thank you for coming out. We had a great turnout. So that is all. Oh, yes. Right here, actually. Thank you. Thank you.
Still eyes.